Welcome to the Hematology 1 review. So we are going to cover kind of a PowerPoint reviewing all of Heme 1 just to kind of get that back into your mind for the final exam. So again, your Heme 2 final exam covers both Heme 1 and Heme 2. And the exam itself must be passed with a 73% or higher in order to pass the class. So again, no matter what grade you have at this point, if you cannot pass your final exam, that tells us then you're still not quite grasping it. There's, of course, always a retake exam. The retake will be a different test, not the exact same test. So just kind of perspective on that. So I think it's really important that we make sure we go back to Heme 1, rediscuss those items, because I'm sure some of it's kind of been forgotten or there's areas in there that you still struggle with. Probably one of the main areas I find most students struggle with are the anemias. They either forget the anemias or they get them confused or whatever they might be. So that's what we will do today. I will also make sure that I send out our Heme 1 final review that we had out of Heme 1. It was on some questions and some pictures, which the pictures were good to help get you ready for the lab practical. Same for this case. On your lab practical in Heme 2, you will have pictures again. So still just as important to be able to know those cells or inclusions or whatever they may be. All right, so I'm going to share my screen. I will have sent all of you the PowerPoint that we are going to use here so that you can follow along with it. So going all the way back to the very beginning of Heme 1, we learned hematopoiesis, and we learned there are 3 phases to fetal hematopoiesis. So how do we make our red blood cells, our white cells, and other cells in a fetus? So it starts with the yolk sac phase, which goes from about 2 to 5 weeks. Then it moves into the liver phase, which we call hepatic, 5 weeks to 6 months. By the 6th month, the bone marrow will become your primary site of hematopoiesis and onward. So keep this timeline in mind. Keep knowing it. We also learned as part of hematopoiesis that everything comes from that stem cell. So on our hematopoiesis chart, all cells generate from that pluripotential stem cell. So stem cells we know are capable of self-renewal. They're capable of reconstituting an irradiated host. So if you wipe somebody's bone marrow clean and you give them stem cells, they're able to make all the cells again because everything ultimately comes from that stem cell. So here's what that hematopoiesis chart looked like again. So there is that pluripotential stem cell on the top, and then it splits into 2 sides, the myeloid side and the lymphoid. The only thing on the lymphoid side, again, is lymphocytes. Everything else exists on the myeloid side. So how we make our red cells, our platelets, and then the rest of the white cells. So you do need to know all these cell lines in here. We've learned them all. We've talked about a lot of these, so hopefully a lot of this is familiar to you, but keep the hematopoiesis chart in mind. So starting with erythropoiesis, we discussed erythropoietin, which is a hormone, absolutely necessary to have it in order to make a red cell. If we did not have this hormone, we're not going to be able to make that red cell. So where is erythropoietin made? I hope this is drilled in by now that it is the kidneys. There are oxygen sensing cells in the kidneys that know when your body is getting low on oxygen, and they will kick out more erythropoietin to go to the bone marrow to get more red cells made. All right, so the actual order of red cell production line goes from stem cell to myeloid to CFU-GEM, then to BFUE, CFUE, and then to pronormoblast. On this, pronormoblast is the very first cell that we can actually see on the microscope, under the microscope. So you do need to know pronormoblast, basophilic, then polychromatic, orthoretic red cell. And you should be able to identify these, especially identify the polychromatic, the orthochromic, the retic, and of course, I hope to God, the red cells. Those 4 are for sure identifiable. Pronormoblast, basophilic, they look similar, but keep in mind, pronormoblast is bigger, has more nucleoli. Basophilic, a little bit smaller, less nucleoli. All right, out of these cells just listed, which one has that pycnotic nucleus? And that would be your orthochromic. So remember, a pycnotic nucleus is that really condensed, dark nucleus where all the chromatin is completely condensed and just looks like one dark solid mass. So that would be a very key sign of an orthochromic, is having that pycnotic nucleus. Which of the following is 1 to 2% in the peripheral blood? And that would be your reticulocyte counts. So we, at any time, have roughly 1 to 2% retics floating in our blood. That's normal. Anytime that goes higher than that, that tells us your bone marrow is responding to something. It's kicking out more retics, maybe because your blood cells are getting hemolyzed or you're losing blood. So whenever your retics go high, it's always an indicator of bone marrow response, which is the next question here. Which cell do we measure to indicate bone marrow response? Retics. So a high retic count tells us your bone marrow is responding. Which cell in this line is last capable of cell division? And that's going to be the polychromatic. So that's the very last one that has mitosis in this cell line. All right, so here's some examples of those cells in picture form that you should be able to identify. So the one on the upper left, this is a polychromatic normoblast. And I know that because, one, the size. If you compare this to the red cells around it, it's not that much bigger than them. It's a little bit bigger, but not much. Also, the cytoplasm is a light blue-gray color. It's not that dark looking. And then the very key is the chromatin inside that nucleus. It's all clumpy. It's the one that I always refer to as checkerboard because it's clumpy, patchy, So it's kind of like that black-white checkerboard look. So those are the key features of a polychromatic normoblast. Over on the upper right here, that's probably the one that's easiest for most people. This is the orthochromic normoblast. This is the one that has the pycnotic nucleus. So this is what I was kind of discussing here, that very dark, completely condensed nucleus. So that is orthochromic. And again, when you look at this cell, it's the same size pretty much as the red cells around it. It's hardly bigger than them. So it's almost completely down to a red cell. And then the one in the bottom middle here is a basophilic normoblast. So again, if you look at this cell size compared to the red cells around it, much bigger. So you know right away by it being so large, it has to either be a pronormoblast or a basophilic because it is so big. The other key thing is how dark blue, or what we call basophilic, the cytoplasm is. So the cytoplasm is really dark blue. And then the key here is when you look in the nucleus, do you see any nucleoli? So again, a pronormoblast has around 1 to 2 nucleoli. A basophilic normoblast has 0 to 1 nucleoli. So in here, it's hard to tell on a picture. I don't really see any. There may be one right there, but I'd have to see it under a microscope, of course. But otherwise, it really doesn't look like there's any nucleoli in this picture. So that is your basophilic normoblast. So upon learning about red cells, we also learned they need energy, of course, because once they get rid of that nucleus, you know, they don't have organelles, it's hard for them to make energy, but they still need to function as a cell. So they have a couple of glycolysis pathways that are able to give them some energy along with other things. So the main pathway to give energy is the anaerobic glycolysis, or what we call Embden-Meyerhof pathway. This will give the cell 95% of where it gets its energy from. At the end of that anaerobic glycolysis pathway, you will get 2 ATP. And again, ATP is always the energy for the cell. That's what energy units are for the cell. The other pathway, called the aerobic glycolysis, is also named the Hexose monophosphate pathway. This pathway is getting us glutathione. Glutathione has a very important role in our body in that it protects our red cells from oxidative injury. So we need that to keep our cells protected. Inside all the red cells, we have hemoglobin molecules. Of course, the function of hemoglobin is to bind oxygen so that it can deliver it to the tissues. The hemoglobin is made of 4 heme groups, 4 globin chains. So 4 and 4 make 1 hemoglobin. Each hemoglobin can carry 4 oxygen. So each one of these heme groups will bind an oxygen. So if there's 4 heme, there's 4 oxygen it can carry. So 1 heme group carries 1 oxygen. The entire hemoglobin protein carries 4 oxygen together. Within the heme group is a protoporphyrin ring. The protoporphyrin ring is made of carbon, nitrogen, and hydrogen molecules, and inside the center of that is iron. And iron has to be in the ferrous state. We want it in the ferrous 2+ state. Otherwise, we're not going to bind and deliver oxygen correctly. So ferrous for us is the same. We want the 2+ state. If it changes into that ferric 3+ state, it's not going to work. That's what we call met hemoglobin. So remember, met hemoglobin was any time your iron did turn into the ferric 3+ state. Normal hemoglobin should always have ferrous 2+ states. So remember, ferric is icky, ferrous is for us. We want the ferrous 2+. Overall, the heme groups are made in the mitochondria. The Globin chains are all those amino acids linked together. Those are made in the ribosomes. Afterwards, they come out, I say they get married in the cytoplasm, and they have triplets. And I say triplets because in all of us, we have 3 normal hemoglobins. We have hemoglobin A, hemoglobin A2, and hemoglobin F. So those are the triplets. Those are the 3 hemoglobins that normal, healthy people have. If you have an abnormal hemoglobin, you're going to have some sort of disease with that then. So, oh, I was expecting something different there. But anyway, moving on, our oxygen dissociation curve. So this is basically telling us how is your body binding oxygen and releasing the oxygen. And there are different things that can happen to the patient to put their oxygen curve either to the left or to the right. So if their body temperature changes or their pH of their blood changes or something like that, it will move their oxygen curve on how it's performing. So anytime your oxygen curve shifts to the left, that means you love oxygen. You really, really like it. You have what we call a high affinity for that oxygen. And so you're going to hang on to it. You're not going to release it. You're going to keep it on the hemoglobin molecule. A shift to the right, right release. You can get rid of it. It wants to release it, wants to get rid of it. So that would be a low affinity for oxygen. So that's kind of the definition of what it means when it shifts one way or the other. Now, the other thing you have to just memorize is what will cause it to shift that way. So pH, I know I'm skipping here, but pH, if you have a high alkaline pH, that will cause a shift to the left. So I always try to remember the L's in alkaline for left. And so then opposite is decreased or acidic pH would shift to the right. Temperature, if your body temperature drops, you will shift to the left. If your body temperature increases, you will shift to the right. 2,3-BPG. This is, first you need to know what 2,3-BPG does, and it will make more sense. So 2,3-BPG is a substance in your body that's all about helping oxygen get released and delivered. So if somebody's really anemic, when you're anemic, you need oxygen. You're low in oxygen. Your body will increase the level of 2,3-BPG in response to being anemic to help release more oxygen. So of course, if 2,3-BPG's whole role is to release oxygen, it's going to cause your shift to the right if you increase that level of it. If you don't have that much 2,3-BPG, it'll shift it to the left because you're keeping it then. You're not releasing it the way it should be. So, and then oxygen levels are down here. Low oxygen will shift to the left, high oxygen shift to the right. I think at one point I even had CO2 on the chart in the original one way back in Heme 1 that you can look at. But the way that it goes is the easiest way to memorize this is if you know your pH. So if I know alkaline, the L's in there go with left, that's high. Then everything else is low for levels. Low temp, low 2,3-BPG, low oxygen. If I know that a low acidic pH is to the right, then everything else is opposite. High temp, high 2,3-BPG, high oxygen. So that's kind of how I memorize it. Just keep in mind left loves, meaning great amount of affinity, right release, low affinity, and then you have to kind of go with the rest of these results. I think in the original PowerPoint when I did this, which was chapter 10, I explained why each one caused this shift. If you want to go back and re-listen to that to know the actual causes, please do so. But basically know your shift, know what 2,3-BPG does, especially if you're anemic, why that would go up to help release the oxygen so it shifts to the right. I'm kind of stumbling on my words right now, but this is always a little bit confusing for people, but just learn what you can. I know this is something that you also see at some point in chemistry as well. So again, there are abnormal forms of hemoglobin. Methemoglobin was when your iron became ferric in the 3+ state, that icky ferric iron. So that would turn into methemoglobin. Self-hemoglobin is just due to different types of drugs that the person might be exposed to or taking. And then carboxyhemoglobin, that is carbon monoxide poisoning. So if your body has a choice between oxygen and carbon monoxide, it will always pick the carbon monoxide. And that's how carbon monoxide poisoning gets deadly, is if you get enough carbon monoxide poisoning in you, your hemoglobins are binding all that carbon monoxide up. What happens then is you're not getting any oxygen to the tissues because it's not picking the oxygen. So that is how it becomes deadly. Again, with carboxyhemoglobin, if somebody has been exposed to carbon monoxide poisoning, this will have a bright cherry red color to the blood. Red cell indices, you have to know your calculations. There was a bunch from Heme 1. So the first set of calculations that we really probably covered was the red cell indices. So this is your MCV, MCH, MCHC. So know each one of these calculations, know the reference ranges for each one, and basically what does it mean. So MCV is indicating your size of red cell. So MCV will tell us the size. And remember, we apply those terms. It might be microcytic, normocytic, or macrocytic, and it just depends on what the MCV result is and where it is in reference range area. So again, if it's less than 80, it'd be microcytic, saying you have small-sized red cells. If it's between 80 to 100, that's normocytic, and above 100 is macrocytic, meaning you have large red cells. MCH is giving us the weight of hemoglobin. There is no terms to really apply here. You just need to know how to calculate it. And then MCHC is giving us the concentration of hemoglobin. And so these are the terms hypochromic if it's less than 32, normochromic if it's between that reference range, and hyperchromic if it's greater than 36. Now, a word of caution, when it's hyperchromic, greater than 36, it should go along with spherocytes. Spherocytes are one of the only true things that can cause a high MCHC count. There are things that happen to the patient's sample that might cause it, like lipemia. If somebody's sample is really turbid lipemic, that will cause a falsely high MCHC, and so we have to always be aware of that. The only really true time we see an accurate MCHC to be high is when there's spherocytes. So you should link spherocytes with a high MCHC at the end of the day. All right, so know these. The other reason that we do these is this is how we categorize our anemias. Every anemia will fit into either a microcytic, normocytic, or macrocytic category. So that's where these become big as well. The minute that we start going into anemia case studies, you should right away be looking at the red cell indices to kind of get narrowed down what type of anemia might even be possible. So there's all the terms applied with the ranges. And then again, here are some of those anemias. Iron deficiency, Thalassemias, Citroblastic, very much microcytic type anemias. You could also add anemia of chronic disease. That would be a microcytic. Normocytic is hemolytic anemias, aplastic anemia, sickle cell anemia would be normocytic, and your anemia of chronic disease actually can fit here too. That's the one that kind of fit in both categories. For macrocytic, the big ones was liver disease and megaloblastic anemia, and then we also added myelodysplastic syndrome, which we just learned in Heme 2 this time. So these are some categories. You can see how just knowing the MCV, MCHC result helps you already narrow down what possible anemia could be existing. RDW was a calculation always done by the analyzer. It's not something that we have to calculate, but basically RDW tells us that we're having variation among red cell sizes. So that is a term called anisocytosis. So it means variation. And again, the reference range for RDW is around 11.5 to 14.5%. All right, so now going into the anemias, there is two classic mechanisms to anemias. Either they're formed because there's ineffective erythropoiesis, in which you're making defective cells, and so they're kind of destroyed before they ever leave the bone marrow. That can result in anemia. Megaloblastic, Citroblastic, Thalassemia are all good examples of an ineffective erythropoiesis mechanism. The other mechanism to forming an anemia is insufficient. You're just not making enough red cells. Iron deficiency would be a great example of that. So those are the two main mechanisms to forming anemias. So again, here are your overall categories. I think these are written a little bit nicer. On the macrocytic one, you could also put liver disease over there if you want. So I didn't list every single thing ever, but these are the main anemias that we actually went through. So you should know each one of these anemias listed here and the main cause of why do they form, what happens. All right, so we'll start to go through these. Again, we're going to briefly go through them. I mean, I don't have time to get into every single one in depth, or we'd be here for a long time. Go back to your PowerPoints, go back to what you can to remember these. Iron deficiency is probably the easiest. It's named for what it is. You're missing iron, you're low in it. And that could be you're bleeding out too much. They're kind of losing blood slowly, like chronically. It could be somebody has a heavy like menstrual cycle, growing children or pregnant women who just have a higher need and not getting enough, or poor diets. Anything like that would cause you to be low in iron. So of course, with anemia, you're always going to see a decreased hemoglobin hematocrit usually. Now, with iron deficiency, to confirm that it is, remember these are the ones that are microcytic hypochromic in iron deficiency. You can confirm it by doing iron studies. You would see a low serum iron, a low ferritin, but a high TIBC. So it's bolded there for a reason. Whereas the next one, anemia of chronic inflammation, this one is you have iron, it's there, but it's being impeded so that you can't really bind it or bring it to the cells. So there are what we call acute phase reactant proteins, APR proteins, that anytime somebody has like a chronic illness that causes them a lot of inflammation, these acute phase reactant proteins elevate during times of inflammation. As a result, they go and bind the iron so it's not available and kind of take it away. So anemia, chronic inflammation, you have some iron available, it's just being bound up and used in different ways. So again, you will see a low serum iron because you just don't have it floating in the iron because it's being bound. Your ferritin is a little tricky on this one. Ferritin can either be low, normal, or high. And the reason for that is ferritin is an acute phase reactant protein, so it does respond to inflammation. And so it can be high because it's responding to that, or it can be normal, you know. So, and then the key is a low TIBC. So that TIBC really helps separate between iron deficiency and anemia of chronic inflammation. Citroblastic is, again, you do have iron, but it cannot get put into the developing red cell. There's two main reasons. One is lead poisoning. So if you have lead poisoning, that lead is kind of interfering with how the cell is getting made and not being able to put the iron in there. And then the other reason would be what we call porphyrias, and you should learn those more in chemistry. Porphyrias mean that you have a disease in which you're missing one of the enzymes to make the protoporphyrin ring inside the heme. If you can't make that ring, you can't put iron in there to make a red cell. So you will see on a bone marrow, if you stain it with Prussian blue stain, again, Prussian blue stain is the stain we use for staining iron. If you were to stain somebody's bone marrow that has sideroblastic anemia, you would see what we call ring sideroblasts. So they would show the iron granules available circling around, waiting to get put into the red cell, into that protoporphyrin ring, but they can't. They're just circling, waiting. And so that's the name ringed sideroblasts. If it is lead poisoning, the other big thing you would see with lead poisoning, again, would be that basophilic stippling. So All right, again, all three of these are microcytic hypochromic category, with the exception that anemia of chronic inflammation can also fit into the normocytic normochromic category. All right, anemia is due to some sort of globin chain defect. So you have thalassemias and sickle cell anemia. We learned these week 1 of this quarter, actually, of Heme 2. So you should have a lecture that you can go back and listen to if you want to. Again, thalassemias are a decreased rate of hemoglobin synthesis, so it's a quantitative issue. You're not making enough of the globin chains, and that's because you're missing genes. And it depends on if it's a, oh my goodness, I just lost the thought. It depends on if it's a heterozygous or homozygous condition, how severe it is, and how many genes that you're missing. So obviously homozygous, much more significant disease, you're missing all the genes together. So as a result, you're just not going to make it. They're going to name the thalassemia according to what genes are affected. So if it's alpha thalassemia, you're not making the alpha chains. If it's beta thalassemia, you're not making the beta chains. Go back to the original PowerPoint, make sure that you know there's categories of each. In beta thalassemia, there was 3 categories, and it basically just went minor, intermediate, major. Beta thal major, of course, is the one that's transfusion dependent, very severe. And then the alpha thalassemias, there's a carrier, there's, I can't remember all the names exactly, carrier trait, I can't remember the second name, Hemoglobin H disease, and then hydrops fatalis. And again, as you go down, it's missing 1 more alpha gene. So in the first one, they're only missing 1 alpha gene. Then they're missing 2. In hemoglobin H, they're missing 3 out of the 4 alpha genes. And then in the last one, hydrops fatalis, they're missing everything, and that's where you get that hemoglobin BART. So definitely go back, review that, because it's a lot of info. But again, thalassemias overall are a microcytic hypochromic anemia. On the other side, we have sickle cell anemia, which is normocytic normochromic, and this one is a qualitative issue. You're making enough globin chains, all of that's being made, but you have altered the amino acid sequence in the globin chain, which changes the hemoglobin molecule. So in the case of sickle cell anemia, glutamic acid is replaced by valine in the 6th position on the beta chain. When it replaces it, it makes an entire new hemoglobin. In this case, we call it hemoglobin S, and that's what forms sickle cell anemia. So there's other ones. There's hemoglobin C disease. You do need to know that amino acid substitution as well. And then hemoglobin E disease would be the other one I would ask you to be more familiar with too. So go back and look at those. Again, both of these are very characteristic to see target cells in presence of both thalassemias and sickle cell anemia. There's lots of other poikilocytes you can look at for each one, but the target cells were a main feature. All right, hemolytic anemia is destruction of the red cells. Again, your bone marrow just can't keep up, and that's what eventually forms anemia. You will definitely see increased reticulocytes. As we discussed, increased retics mean your bone marrow is trying to respond to something. So anytime the retics are up, it's usually a sign of hemolysis occurring. There are a lot of reasons why you would have a hemolytic anemia, and we have chapters upon chapters, or maybe not chapters, but PowerPoints, many PowerPoints. We hit like 3 weeks of hemolytic anemias there at one point. Aplastic anemia is bone marrow failure, and most of the time we don't know why it happens, but your bone marrow can fail and all of a sudden just not be producing cells. Both of these are under the normocytic normochromic category. All right, so here are some key findings of hemolysis. Remember, hemolysis we separate into 2 categories, intravascular and extravascular. In other words, it just means where is it occurring. If the hemolysis is occurring inside the blood vessel, then it's intravascular. If it's occurring in the spleen, then it's extravascular. So key findings, in intravascular you get schistocytes, which are those fragmented red cells. You will see hemoglobinemia, which means free-floating hemoglobin in the blood. Well, that makes sense. If your red cells are getting destroyed inside the blood and breaking open, they're going to release that hemoglobin, and we're going to see hemoglobin molecules floating freely in there called hemoglobinemia. Hemoglobinuria means hemoglobin in the urine, and that's because upon hemolysis, that hemoglobin that comes out and is floating freely gets filtered through the kidneys and passes into the urine. Hemostaterinuria is, hemostatirin again is a type of iron that can also go into the urine upon hemolysis. Decreased haptoglobin. Haptoglobin is one of those proteins that tries to bind the hemoglobin as it's coming out of the red cells to save it so it does not get filtered by the kidneys. So it wants to save the hemoglobin, bring it to the liver so that we can reuse the parts of it. But when it does this, haptoglobin gets depleted in the process and doesn't come back. So anytime there is an intravascular hemolysis, your haptoglobin will get used up by trying to save the hemoglobin and bring it to the liver. And then in extravascular, this one is spherocytes, which are, again, a red cell that has reduced surface area, but just as much a volume on the inside. So they're rigid, kind of formed little spheres or balls. And when they go through the spleen, they can't make it through the little small crevices because they're not flexible. They're very rigid. And so that will cause them to burst, and that's always a sign of an extravascular hemolysis. In both, you will definitely see increased bilirubin because remember, as you go through those pathways to reuse the parts of the hemoglobin, like to reuse the iron inside it, you reuse the amino acids, all that, bilirubin is a waste product of your hemoglobin breaking down. Your protoporphyrin ring will become biliveridin and then become bilirubin. So anytime somebody has a lot of hemolysis occurring, you will see increased levels of bilirubin as a result because you're increased in the amount that you're breaking down. And so that usually leads to jaundice, that yellow coloring to the skin and eyes. You'd also see increased LDH. LDH is an enzyme, lactate dehydrogenase, and maybe you guys have heard of this in chemistry, but LDH is found a lot on the inside of cells. So as your cells are hemolyzing, they're letting out everything that's inside them, including LDH. So you'll see higher levels of LDH because they've been released out of the cells. Increased reticulocytes, because again, your bone marrow is going to try to replace any of these cells that are getting hemolyzed. So they're going to kick out retics just to get some cells out there. And then decreased A1c. This one's a little trickier, but again, if you're in chemistry, you should know hemoglobin A1c is always something that we use to monitor diabetes. It tells us somebody's glucose levels over the last like 3 to 4 months. Well, in the case of hemolysis, if that's supposed to be an indicator of a 120-day lifespan of a hemoglobin, if you're having hemolysis and breaking down hemoglobin faster than normal, you're going to have a lower A1c result because it's not living the 120 days that it normally should. So it'll show a low number because it's not getting to the 120-day mark. All right, so some of the hemolytic diseases we've learned, MAHAs, we've kind of been bringing MAHAs back up with the COAG unit, but again, the whole principle of the MAHA group is that it's based on clots happening in the smaller vessels like capillaries and arterioles, so that's the microangiopathic. So when these little clots are formed in those blood vessels, as blood is trying to get by there, the clot will shred the red cells, and that's the hemolytic parts. So it is a group of disorders. HUS, TTP, and DIC, all three belong in the MAHA group. They're all three having clots and causing shredding of red cells. HUS, again, the big thing with this one is it's usually stemming from a food poisoning case due to the toxins that kind of cause all the issues. So like E. coli 157 or Shigella are two big food poisoning bacteria that release some nasty toxins. And so the big thing here is very severe renal failure that kind of goes along with it. It really affects the kidneys. TTP is clots also happening, the same thing, MAHA thing happening. It does have some renal failure, but not as severe. Low platelet counts, and then this one is the one that can also have neurologic abnormalities. Some of those clots might be up in the brain, kind of leading to neurologic impairment. And then DIC, we just discussed this in the COAG unit. DIC is secondary to some other condition in the patient happening, leukemia, sepsis, whatever it might be. And so upon forming these clots, at first it's nothing bad, and then all of a sudden you form so many, it results in one big hemorrhage and very emergency type situation. So we just discussed DAC. I'm not going to go into that. You can revisit the coag unit. So again, it talks about the fragmentation of red cells occurs. Our red cells pass through fibrin deposits in those arterioles and capillaries. So again, that's the principle of what MAHA is. Of course, this is an intravascular hemolysis. It's occurring inside the blood vessels, so you'll see those intravascular signs. So schistocytes, helmet cells. Helmet cells or keratocytes always go along with schistocytes. They're seen in the same things. Malaria was another hemolytic disease learned. So malaria is a parasite that loves to live inside the red cells and use it to make more of itself. But when it does that, it hemolyzes the red cell when it's all done with it. So that's the hemolytic part. So we had 4 species of parasite. You have Plasmodium vivax and ovale, Plasmodium malariae, and Plasmodium falciparum. Plasmodium vivax and Plasmodium ovale usually tend to infect the younger red blood cells, like retics and young red cells. Plasmodium malariae tends to infect the older cells. And it has that really characteristic band trophozoite stage to it. And then Plasmodium falciparum will infect all types of red blood cells, and that's what makes it the more severe form of malaria, because it will infect all of them. And in the Plasmodium falciparum stages, it has a couple unique characteristics. In the ring stage, this is the one that is capable of having 2 chromatin dots. All the other 3 that when they're ring stage just have the 1 chromatin dot. And then in the gametocyte stage, Plasmodium falciparum is very unique in that it gets that crescent or banana-shaped gametocyte stage. All right, the thing with malaria is that's commonly heard is that you will get these intermittent fever and chills. In other words, they don't have fever and chills every day straight the entire time. It kind of goes in cycles. They'll get them and then they'll go away, and then they'll get the fever and chills and go away. And that's because they're getting the fever and chills anytime the actual hemolysis of the red cells is happening. So once the parasites are done using the red cell to grow and multiply, it'll hemolyze it, and then they'll go to another red cell. When that hemolysis occurs, that's when the patient feels the fever and chills. Overall, very brief life cycle of malaria, which you need to know the life cycle. Be sure to go back and visit it. But the overall life cycle of malaria is you get a mosquito bite. It will basically inject into you the parasite. The parasite goes to the liver and lives in the liver cells for a little bit and undergoes, its reproduction process. When it's done there, then it goes into the blood and infects your red cells. Goes more reproduction, goes through the different stages of it, like ring stage, trophozoite stage, that kind of thing. And then upon using that, it will lyse the red cells and go to another red cell and keep doing that over and over. Now, some of the parasite will become gametocytes, and gametocyte stage is the mature male and female stage. Those gametocytes will hang out in the blood. They won't necessarily be directly in the red cells. They'll hang out in the blood. And when the mosquito bites you again and sucks up blood, it sucks back up those gametocytes. Those gametocytes go down to the stomach of the mosquito. They make a little oocyst, which will then reproduce and get bigger, or not reproduce, will mature, and then they will go back up to the salivary glands of the mosquito and wait for another bite. So that's a very simplistic based life cycle. Again, go back to the original PowerPoint and go through that life cycle and know the order of it. I didn't mention this before, but now that I'm thinking about it, you should also know the order of the pathway for intravascular and extravascular, the pathway steps on those. Do know that as well. All right, and the only other question on this slide was what's another parasite found inside red cells that can cause hemolysis? And that would be Babesia. And Babesia, again, is passed by a tick. All right, we had some hereditary conditions that resulted in hemolytic disease. Hereditary spherocytosis, this is due to a protein abnormality in the membrane. And so as a result, it causes the membrane to shed off, and then it reseals itself, making it a spherocyte. Because again, a spherocyte is lesser surface, just as much volume on the inside. And so when you are born with this and you have these shutting off of the membrane, it just keeps resealing itself. It didn't lose anything on the inside, it's just losing the surface. So it makes it a very rigid little sphere, and which will cause hemolysis in the spleen because it can't get through those small spaces. So hereditary spherocytosis, because you have spherocytes, again, you'll see that high MCHC present. The test that's really key to diagnosing this and knowing that it's truly spherocytes is osmotic fragility. So in that osmotic fragility, they take the patient's cells and they add, basically you can just say they add water. And normal red cells can have so much water added, they can take a little bit because they're not that full before they burst. Well, with spherocytes, because they're so full already and they have no room, they're really rigid, they're full of stuff, if you try to add anything to them, they burst really easily. And so that's what it means by increased fragility. They're fragile, they burst quicker. So that's osmotic fragility for hereditary spherocytosis. Hereditary elliptocytosis, this is obviously the way it sounds. You have a lot of elliptocytes. It's another membrane abnormality. You don't need to know which one. Again, in order to know it's elliptocytosis, you need to have at least 30% of your red cells be elliptocytic in shape. Elliptocytes can go with a lot of different diseases. We can see them in iron deficiency anemia. We can see them in thalassemia. But in order to consider it hereditary elliptocytosis, you need to see greater than 30% be that shape. G6pd deficiency is when you are missing the enzyme glucose 6-phosphate dehydrogenase. So that enzyme is a very key place in the hexose monophosphate pathway. So remember, the hexose monophosphate pathway is the one that gave us glutathione to protect our red cells. Well, if you are missing this enzyme, you're not going to make glutathione, and your cells are very risk of oxidative injury. So if that person was to be put on any sort of medicine or drugs that can cause oxidative damage to the red cells, that's what will cause their hemolysis. So they don't necessarily have hemolysis all the time. It's just when they are put on something that can cause injury to their cells because they're missing that glutathione to protect it. A key finding in G6pd deficiency is Heinz bodies. Those are the little inclusions that are made of denatured hemoglobin. So you'll get these little hemoglobin inclusions inside there. That's a key sign of G6pd deficiency. Again, in order to see the Heinz bodies, you have to use supravital stain. You cannot see them on normal right stain, so keep that in mind. The other thing you would see with G6pd deficiency as a result is bite cells. Bite cells are what they look like. They look like somebody literally took a bite out of the red cell. And what happens is when these red cells go through the spleen with the Heinz bodies in them, macrophages in the spleen are screening for that. They look for it, they see, oh, that's a Heinz body, that's abnormal, and they bite it out of the cell to remove that abnormality. So you'll see both the Heinz bodies and bite cells as a result. PNH is paroxysmal nocturnal hemoglobinuria. This one is not necessarily hereditary. It is an acquired defect. So it probably shouldn't have put it under the hereditary label at the top there. The reason I did is we did discuss it in the same PowerPoint as a lot of these. But again, PNH is actually acquired membrane defect, not hereditary. So upon having this red cell membrane defect, basically it looks abnormal and your body targets it for hemolysis. And so these cells are very, what we call susceptible to complement. Complement is a big thing with our immune system. What complement will do is destroy whatever it sees as abnormal or foreign. You know, usually we want it for any sort of like viruses and bacteria and things like that. But if with this red cell membrane defect, it can see this cells as foreign. So it will just set out and destroy and hemolyze these red cells, which is what makes it a hemolytic disease. There were two tests that we talked about for screening and confirming PNH. Sucrose hemolysis, which is sugar water, and then Hem test, which is also known as serum lysis. So know that those are the two tests that go with PNH. And again, it's an acquired red cell membrane defect. With aplastic anemia, there was a bunch of different types of bone marrow failure type stuff. Some belong to the aplastic anemia, others were just bone marrow failure stuff. Again, aplastic anemia overall is a unknown thing that causes idiopathic, I should say. Most of the time we're idiopathic. There is a inherited form of aplastic anemia, though, called Fanconi's anemia, and that one some key signs of that: skin pigmentation, short stature, hypogonadism. So that one is inherited, so we do have a link for where that comes from. Most of the time, aplastic anemia, we're not sure why somebody all of a sudden results in bone marrow failure. Other types of bone marrow failure have congenital dyserythropoietic anemia. So you get these really weird, bizarre red cells. It only affects the red cell line, which is why it's called dyserythropoietic. So again, it's dysfunctional, weird-looking red cells. Dyskeratosis congenita, this is one that has skin pigmentation, dystrophic nails as some key characteristics with it. I always remember the carrot as keratin with your nails. That's how I put that one together. Pure red cell aplasia sounds just like it is. It's only affecting the red cell line. So you will see a decrease in this red cell line. There is an inherited version called diamond black fan, and it's inherited because the stem cell became defective in it, leading to that whole red cell decrease. Myelophystic anemia is when you have bad cells coming in and replacing the good ones. So, and then anemia of chronic renal insufficiency. If your kidneys are not functioning right and they're not making erythropoietin, you're not going to make red cells. So no EPO, no red cells. So know the key features of each one of these bone marrow failure type syndromes. And then megaloblastic anemia, one of our macrocytic anemias. We didn't really have much macrocytic anemia. This was the main guy for macrocytic. It's actually a category of two different types. It can either be due to vitamin B12 deficiency, which there is a form of that called pernicious anemia, or it can be due to folate deficiency. So the deal with these vitamin B12 and folate, both of those are needed to help make DNA for our cells. So if you get really deficient and low enough that you're not going to really make DNA, you're not going to make cells. And so that's where the ineffective erythropoiesis comes in. You're just not making cells at all. And that's why we would see pancytopenia. All cell lines are low because you're not making DNA to make them. So again, macrocytic type anemia, you will see oval macrocytes, hypersegmented neutrophils. That was really unique to just megaloblastic. You should always link hypersegmented with megaloblastic. Okay, finally going on to some other cell stuff. The leukopoiesis, so this is just white cell development. Again, you have your two lymphocytes, T and B cells. In your granulocytes, those are neutrophils, basophils, eosinophils, and then of course you have monocytes. Keep knowing your neutrophil line from stem cell all the way down to segmented neutrophil. It's listed out there for you. For some questions here, which of these is the last cell capable of mitosis? And this would be the myelocyte stage. So myelocyte is the last cell capable of mitosis. Primary granules begin being made in the myeloblast. When do secondary granules begin being made? And that is the myelocyte stage as well. So primary in the myeloblast stage, secondary will start being made in the myelocyte stage. Which cell has a slight indentation? That is a very key characteristic, and that is metamyelocyte. So that's when the nucleus goes from being perfectly round to being slightly indented, because remember, we got to get it to the segment where it's all weirdly lobed and stuff. So in the metamyelocyte stage is the very first one where you will see that nucleus start to become indented. It's kind of like a kidney bean shape almost. And then again, the main function of neutrophils, of course, is phagocytosis, to fight off especially bacterial infections. Other granulocytes, eosinophils, we see these increased in allergies, parasitic infections, and CML. So we just learned CML this quarter, and so you should see high eosinophils in that. What color are the characteristic granules? Again, eosinophils have those beautiful, bright red-orange granules. Charcoal Leyden crystals, this is something we learned a long time ago, but basically when you're looking at a diff, and sometimes you come across an eosinophil that looks burst almost, you see the red-orange granules laying there, and you kind of see a nucleus off to the side, that's kind of charcoal Leyden crystals. Basophils have that really dark purple granule color, and again, those granules will cover the entire cell. That's a key feature of basophils, where the granules cover the cell, so you don't always get to see the nucleus, so they're dark purple. Inside the granules, they have histamine and heparin. And then basophils, which also can become a mast cell in the tissues, will have a receptor for antibody E, IgE. And IgE does have a role with allergies. So does histamine, has a big role with allergies. So they kind of play a role there. So that, and then I already answered the last question. What are they called in the tissues? Mast cells. Lymphocytes' main function is, again, immune system. It's a huge part of the immune system. And when you guys get into immunology, you'll learn more of that. But T cells play a big role with what we call cell-mediated immunity. And then B cells are the ones that can make antibodies. Both B cells and plasma cells make our antibodies. So those are the two that make those antibodies. And where are T and B cells differentiated? Again, T cells become a T cell in the thymus. B cells become a B cell in the bone marrow. Don't you wish everything was that easy? Megakaryopoiesis, so this is the process of making our platelets. We need thrombopoietin hormone in order to make these platelets. So TPO is made in the liver. Inside the platelets, we have granules, and we just went over this again in coag unit, but alpha granules and dense bodies or dense granules. Alpha granules will contain fibrinogen, platelet factor 4, von Willebrand factor, a growth factor. They have a bunch of stuff. And when you are missing your alpha granules, you call that gray platelet syndrome. Dense granules will contain serotonin, calcium, and ADP. And when you're missing those, it's called a storage pool disorder. And again, got to know your calculations. Make sure you know these following calculations for your final exam from Heme 1. All right, so that is a nice overview of Heme 1. And I will send out some other review material as well to help you. If you guys have any questions, as always, please feel free to email me. I wish you guys the best of luck. Thank you.